prepared and for the division. Okay. So during this stage, the cell undergoes to normal growth process while also preparing the cell division. So synthesis of energy-rich compounds, proteins, and replication of the DNA result in increased the volume of nuclear and cytoplasmic contents. Therefore, this phase is known as a preparatory phases. Preparatory means initial phase. This one to ready to division. So that's why all the requirements are contained and accumulated in the nucleus cytoplasm. So all the requirements means proteins, enzymes, and other compounds, biological compounds, all uh, are accumulated in the cell. So that's why this interphase we call as the preparatory phase. In the interphase is the longest phase in the life. There is cell cycle. It is consist of the G1 phase, S phase, G2 phase, and mitotic phase. So G1 phase means gap period 1 and synthesis phase, and G2 means gap period 2 and mitotic phase. So here the G1 phase. G1 phase means gap period 1. So the young daughter cell grows in size during this period. In this phase, the RNA, proteins, and enzymes essential for the DNA synthesis are formed in the cell. So all types of the means enzymes, proteins, essential biochemical compounds, all are they are accumulated in the cell for the DNA synthesis. So in this stage, the cell is ready to synthesize the DNA. Okay. So then second subphase here, S phase. S phase means synthesis phase, synthesis of the DNA. The replication or synthesis of DNA occurs during this phase. At the end of this phase, duplication of the DNA takes place in the cell. So in this phase, DNA is synthesized. And G2 phase means gap period 2. During this period, the cell continues to grow and synthesis new proteins. Synthesis of nuclear RNA, ribosomal RNA, and messenger RNA occur. Okay. So the latter, G stands for the gap period. We already discussed that one, and is followed for the M phase, means mitotic or meiotic phase. Similarly, here also they are synthesis the molecular or nuclear RNA, ribosomal RNA, messenger RNA occur. Okay. Then M phase means mitotic phase. So in this mitotic phase, here there are the so here mitotic phase again divided into four stages are there: prophase, metaphase, anaphase, and telophase. Okay. So mitosis is division of cell into two daughter cells which are identical and contain the same number of chromosomes, which are the similar to the parental cells. Though the mitosis occurs in somatic cells and occur in the both haploid and diploid cells also. Only the mitosis occur only the somatic means vegetative cells are there. So both the daughter nuclei and mother chromosomes all are the similar in number. Mitosis occurs in somatic cells and also haploid and diploid cells. So the active mitosis division is observed in the meristematic and apical and later meristematic tissue. Mitosis was first observed by the Strasburger in plants. So mitosis means Fleming. Fleming is the first to introduce the word mitosis. Okay. So the mitosis, the process of mitosis is characterized by the duplication of the chromosomes. Their separation and distribution to the daughter nuclei. So this part of the cell division is called as a nuclear division or karyokinesis. So nuclear is the divide that process is karyokinesis. This is the followed by the division of cell cytoplasm to form the two daughter cells, which is called as a cytokinesis. Okay. What is the karyokinesis? Here, the process of karyokinesis involves the division of nuclei. You know the process. So nucleus divide and after the cytokinesis is divide. In this karyokinesis, here four stages are there. That is the prophase, metaphase, anaphase, and telophase. So prophase means it is the first phase of the mitosis. So during this phase, nucleus became the spherical in shape. In early prophase, so see here, this one is the prophase. This one, okay, sorry. So prophase, this is the prophase stage of the nucleus cell. So in early phase, the chromatin is spread out the a network-like structure. Gradually, the chromosome became a thick and condensed in condition and form two chromatids, which are joined together at the center. That one we call as a centromere. Center of the chromosomes we call as a centromere. During the early prophase, the chromosomes are evenly distributed. As the prophase progress, the chromosomes move towards the nuclear envelope. So the nucleolus and nuclear envelope disappear at the end of the prophase. Means late prophase, nuclear membrane and nucleolus both are the gradually disappear at the last stage of the prophase. So then after the prophase, the cell enter into the metaphase. So in this chromosomes are distinct. The early visible due to the condensation of the chromosome. So a new structure, the spindle fibrous, appears in the cytoplasm, which is oriented longitudinally between the two poles. Okay. Here, the fibrous of the spindle that is extend from one pole to the spindle fiber is the kinetochore of the chromosome called as the chromosomal fibrous. The spindle fibrous we call as the chromosomal fibrous are there. So here the chromosomal fibrous and fibers that are extend from the one pole of the spindle to other pole are called as the continuously fibrous are there. Okay. So here the chromosomes get attached to the spindle fibrous with their kinetochore and their arms are freely in the cytoplasm. These chromosomes move towards the equatorial plate and get arranged in the equatorial plane. So That's why it we call as a equatorial plate. That one we call as equatorial plate or metaphasic plate. So this you see here, this one, all the chromosomes are arranged in the center. This metaphase. This is the equatorial plate. All are arranged in the center of the cell. Uh, that one metaphase. Okay. So, so next one is the anaphase. In this centromere of each chromosome divide longitudinally and allows the separation of two sister chromatids as the spindle fibers begin to contract. The sister chromosomes start moving towards the pole. So during the movement of the chromosomes to the poles, the centromere leaves ahead and arms tail behind to the. They are to assume a different shape of the chromosomes are there. That is the in V shape. That one we call as a metacentric. Okay, that one we call as a metacentric, and some L-shaped chromosomes are appear. That is the submetacentric and acrocentric or telocentric. So acrocentric is looks like a J-like structure, and telocentric it is the I-like shape. As the spindle fibers begin to contract, the sister chromosome start moving the towards the pole. During the movement of the chromosomes to the pole, the centromere leaves ahead and arm tails. That is it. At the end of the anaphase, two sets of the daughter chromosomes. These are reach the opposite poles and form the densely packed group of two poles are there. Like this here. So telophase. The changes that occur in the telophase are almost reverse to the prophase. The daughter chromosomes that are arrived at the pole begins to nuclei and form chromatids. The nucleus and nuclear envelope repairs and surrender the chromosomes. The spindle fibers completely disappear. At the end of the telophase, two daughter nuclei are formed. with the same number of chromosomes are uh, parent nucleus. So here both daughter nucleus are formed. Okay, cytokinesis. Now in the telophase, what happens here? The chromosomes are they are the pulled into the two opposite or edges of the cell. Another one is the cytokinesis. See here, this is the cytokinesis. See, this one is the cytokinesis. So in this cytokinesis, divide the cell cytoplasm into two separate cells. Okay, two separate cells are in the plant cells. It is the accompanied by the formation of the pragmoplast and cell plates at the end of the karyokinesis. So after the formation of daughter nuclei, the remnant of the spindle apertures at the equatorial region form the barrel-shaped structure which is known as a pragmoplast. So these pragmoplasts are accumulated at the center of the uh, cell. Means the vesicles of the Golgi complex also accumulated along the formoplast, the pragmoplast and form the cell plate. The cell plates grows centrifugally, dividing the cytoplasm into the two plots. The cell plate undergoes to chemical changes to form the middle lamellae. On the both sides of the middle lamellae, cytoplasm secrete the primary wall, resulting the formation of the two daughter cells. Sequence of mitosis here. In this mitosis, the constant chromosome. They are maintain the constant number in chromosomes as well as mother cells and daughter cells. Okay. So the daughter cells formed by the mitosis resemble their parent cells in all the characters, means morphological and chemical and physiological and functional also. So this mitosis helps in the replacing the old damaged cells with the new cells. This is also so this mitosis is replaced the damaged cells. So in unicellular organisms, mitosis helps in the reproduction. Okay. So this is the mitosis process. Mitosis means so the mother cell undergoes through division and to produce the two equal cells in the chromosome number also in equal. So that is the mitosis. Then meiosis. What is meiosis here? The meiosis was first was described by the Oscar Harting. Oscar Harting is the first who introduced this one. The sea urchin egg. So the term meiosis was coined by the J.B. Farmer and J.S. Moore in 1905. It is a special cell division that occur only in the applied reproductive cell and chromosome number is reduced to half. So hence it is called as a reduction division. Why here the chromosome number is to reduce it in their number. So that's why, for example, 10 chromosomes are there in the mother cell and the daughter cells they have the 5. Means half of the chromosomes are divided and to produce the two daughter cells. In plants, it takes place in the microspores and megaspore mother cells. Microspore mother cells undergoes to the reduction division to produce the microspore. Haploid microspores are produced. Then megasporogenous or megaspore mother cell undergoes to these reduction division to produce the uh, four megaspore. These are also haploid conditions. So process of the meiosis. So meiosis there consists the two cell divisions. They are the takes place one after other. Means meiosis one and meiosis two. So the first cell division is diploid parent cell in and undergoes to the division to produce the two haploid daughter nuclei. This type of the division is first meiotic division. Okay. Okay. So in this second meiotic division, the formed two haploid daughter cells again divide mitotically and produce four haploid daughter cells. Okay. This division is called as a second meiotic division. The cell division is in all aspects similar to the mitosis division. The meiosis consists of two successive nuclear division, that is the meiosis one, which is result to produce the two haploid cells, and the meiosis two. These two haploid cells became mitotically produced the four cells. So that is the meiosis one. It is the in meiosis. Similarly, here prophase, metaphase, anaphase, and telophase are there. See here. Prophase one. It is the longest phase of the meiosis. It takes place about 12 to 24 hours to complete this phase. This phase is further divided into five subphases like leptotin and zygotin, packetin, diplotin, and diatin. So prophase is the first phase of the meiosis one. It takes long time, around 1 day, means 12 to 24 hours it takes. And these again divided into five subdivisions are the substages are the, that is the leptotin, zygotin, packetin, diplotin, diatin. So leptotin means it is the first phase of the prophase and very shortest phase of the prophase. Okay. In the leptotin, the chromosomes appear is very long, narrow threads. These chromosomes consist of a bread-like, that is the structure is called as a chromosome. In this stage, the nucleus is very distinct. The nucleus contains a set of chromosomes, uh, one divided from male and another from female gametes. These are the known as homologous chromosomes. Uh, Another one is the zygote. See here the diagram, the picture of the zygote here. The chromosomes became short and denser in condition. Here two homologous chromosomes, one being derived from female and other from the male parent. So here start the pairing with each other This processing of the pairing is called as a synopsis, which is the facilitated by the protein called uh, synoptomimal complex. Synoptomimal complex. So once the synopsis complete, the homologous pair is called as a bivalent. The number of the bivalent are equal to the half of the number of the diploid chromosomes. So the pairing may occur in the three ways are there. In proterminal synopsis, it means the pairing of the homologous chromosomes became at the both ends and progress towards their centromere. So procentric synopsis means the pairing of the at the centromere and progress towards the end. Random synopsis or irregular synopsis. This one also the pairing starts various points of the chromosome, which are the homologous chromosomes and simultaneously. Another one is the packaging. So here, which are the form the bivalents we can clearly observed in the packaging to do the condensation of the chromosome. The chromosomes are clearly observed in the packaging stage. Each homologous chromosome splits um, length wise to form the two sister chromatids. As a result, each bivalent appears as a four standard structure called as a packed in tetrars. So the different chromatids present in packet in tetrads known as the non-sister chromatids. Okay. Um, um, in this stage, the homologous chromosomes remain, which are the remain contact to other at the certain points. These are called as a chiasmata. Chiasmata means these points the chromosome breaks into small segments. The breakage end and remain reunion result to interchange the chromosomal segments between the non-sister chromatids. This one called as a crossing over. Means this process of exchange of the genetic material between the non-sister chromatids, that one we call as a crossing over. Okay. So of the two newly formed chromatids in each chromosome, one represent the original parental complement and other one is the recombinant. So another one is the diplotin. See here the diplotin. Type it in here. So this is the diplotin. See. So during this phase, the two homologous chromosomes are bivalent, being repel on other and separate from each other due to the weakening of the synaptic forces, except at the region of the chiasmata. So the chiasma are fully visible at this stage and moves from the centromere to the end of the chromosome. This movement of the chiasma is called as a terminationalization, and is done denotes and end of the synapsis of the chromosome. The chromatids undergo condensation and contrast thickening and distinctly appear as the bivalent structure. Next one is the diakinesis. The diakinesis is the final step of the prophase. The chromosomes are at their most condensed from during the diakinesis. The homologous chromosomes in a bivalent are apart from each other, still connected by the at least one chiasma. The result in this setting the to cell to make way away from the metaphase. So the nucleus disappear, and the nuclear envelope is disintegrated. So another one is the metaphase one. In this phase, the spindle structure are formed. The bivalent arranged themselves at the equator or equatorial stage of the cell. The fibers of the spindle get attached to the centromere of the bivalent. The centromere of each chromosome is the point towards the opposite pole. So anaphase. The homologous chromosomes separate from each other due to the shortening of the spindle fiber. Each chromosome with its two chromatids and undivided centromere moves towards the opposite pole. As a result, the two genomes are separated. This process of the separation is known as a disjunction. Thus, reduction is the number of chromosomes occur at this stage. Telophase, the homologous chromosomes pair complete their migration towards two opposite poles, resulting in the haploid set of the chromosomes are formed. A nuclear envelope reform around each chromosome set. The chromosome start became deconcated and uncoiled and became the elongated in some cells. This step is skipped. So the meiosis. So after the karyokinesis, cytokinesis usually occur. See here, this one will be cytokinesis. See here, cytokinesis as the same to the telophase, forming two haploid daughter cells. In some plant cells, cytokinesis is delayed until both the meiotic divisions are complete. Next one is the meiosis 2. The second meiotic division is similar to the mitosis, but the DNA is not duplicated in this interface. The meiosis 2 include four stages are there: prophase, metaphase, anaphase, and telophase. So the prophase, it is a very short phase. The chromosomes with the two chromatids became short and thick. The nucleolus and nuclear membrane disappear. Spindle formation or start. Another one is the metaphase. Each chromosome arranged itself at the equator of the cell. Chromosomes lie in such a way that their centromere remains along the equator and arms extend outward. The centromere divides and two chromatids of each chromosome separate. The daughter chromosomes move towards the opposite pole and as anaphase, anaphase 1. So the chromosomes appear in the different shapes like V, J, I, and L, like that structures are appear. Another one, the telophase 2. In this nuclear envelope forms around each set of the chromosome. Cytokinesis takes place, producing four daughter cells. each within the haploid set of the chromosome. And because of the crossing over, some chromosomes are seen in have the combined segments of the original parental chromosome. The nucleus and nuclear membrane is reappearance in this telophase too. Another one is the cytokinesis too. So karyokinesis followed by the division of cytoplasm. In plant cells, the wall is laid down, resulting in the formation of four haploid cells. Here, the significance of the meiosis. The meiosis helps in the maintenance of fixed number of chromosomes from generation to next generation, generation to another generation. It helps in the formation of the gametes and sexual reproduction. The crossing over helps in the production of the recombination, which results generation of the gametic diversity. That is the genetic diversity is occur. These variations of these bases help in the origin of the new species and evaluation during the meiosis chromosome assert independently resulting in the variation of the progeny. So cell cycle regulations, which are the involved in the start of the cell cycle. The main function of the cell cycle it is to duplicate and gene of divide it equally between two daughter cells. So for this to happen, cell cycle should proceed in the correct order, and each stage of the cell cycle is to become a completed before next commences. So if the metastasis is blocked and the cell arrested at the metaphase until the block is removed. Similarly, if DNA replication is prevented, the cell does not attempt to undergo mitosis because mitosis is dependent upon the completion of DNA replication. Each eukaryotic cell possesses a number of regulatory mechanisms to ensure the proper division of the cell and inhibited subsequent stages. In case of the failure, these regulatory mechanisms are called as a cell cycle checkpoints. The checkpoints present in the cell cycle of these three stages are there, that is the G1 checkpoint, G2 checkpoint, and M. So G1 checkpoint here we start the starting point, east and restriction point in animal cells. The DNA damage and other external factors like size and cell are evaluated at the G1 checkpoint. If the conditions are unfavorable, the cell is not allowed to enter the F phase. Once the cell possesses this checkpoint, it enters in S phase and undergoes DNA replication and cell complete cell division. G2 checkpoint. The second checkpoint exists in G2 stage before the cell enter into the cell division. G2 checkpoint ensure where all the chromosomes are replicated and the replication DNA are not damaged. It is also concerned with the condensation of the chromatin, which is the procedure mitosis. The chromatin condensation is associated with the phosphorylation of the H1 histone protein. The G2 checkpoint is associated with the interrelationship between the H1 phosphorylation and chromatin condensation. Okay. So in M checkpoint, it occurs at the end of the metaphase stage of the mitosis. It is also known as a spindle checkpoint. This checkpoint ensures The sister chromatids are attached to the spindle fibers before the cell enter anaphase stage. It also prevent the separation of the duplication chromosomes. So until they are they are attached to the spindle aperture apparatus. So molecularization is the basis of the cell here. Um, the cell is controlled by the regulatory molecule that either promote entry of the cell to next phase or inhibited the cycle. These important molecular that control the cell cycle are cyclins and what is the enzymes means proteins and enzymes are there. Cyclins are proteins that have no enzymatic activity of their own but activate by binding to them. So all cyclins are named according to the stages at the assemblage with the CDKs. Common classes of the cyclins include G1 phase cyclins, G1 or S phase cyclins, and S phase cyclins, and M phase cyclins are there. So this is the cell cycle and cell division. So here summary: a new daughter that is the a new daughter cell is produced by the division of the pre-existing cell. The division of the cell nucleus that includes to divide the nucleus division that one we call as a karyokinesis and the division of the cytoplasm that we call as a cytokinesis. Generally, the karyokinesis followed by the cytokinesis. The process of cell division commonly occur by two methods are there, that is the direct cell division or indirect cell division. So direct cell division means they are to produce binary fission means amitosis division that we call as means producing daughter cells are size there is no equal in size. Meiotic division, so mitotic division, so there are mitotic division there are the identically similar in the mother cell. In meiosis, there are the four haploid nucleus are produced from the diploid mother cell. Here, prophase, metaphase, anaphase, and telophase is also there. The newly formed cell are attained a specific size and then is divided. These series of events that occur being two successive cell division called as a cell cycle. Okay. Huh?